St. Philomena's Public School, Ilangi. Today I am going to discuss about a project that I dealt with, biogas plants. In this project, I am in a search for the most efficient substrate in biogas production.
This is the typical structure of a biogas plant. This we see here is called the inlet, where a mixture of organic waste and water are added. This mixture of organic waste and water is called slurry. The slurry is then conveyed through the pipes into a large tank 10 to 15 feet deep called the digester tank. The digester tank hosts some anaerobic bacteria. These bacteria are methanogens, acidogens, etc. They digest the cellulosic material anaerobically and yields biogas. The biogas which is formed is let out through a primary outlet. This primary outlet is connected to pipes which are laid onto different homes, and the biogas is supplied hence. The biogas thus reaching homes is used as fuel for cooking and lighting. The remnants or the residual matter of the slurry gets pushed out through this site and reaches a secondary outlet through pipes. This pushing out of the spent slurry is due to the excessive biogas production. The spent slurry ultimately reaches the site, the secondary outlet site, and is deposited. This spent slurry obtained is rich in nutrients and could possibly replace the use of NPK fertilizers as they support optimum plant growth. So, what are the requirements or necessary prerequisites for the project? The project deals with biogas plants, and so we require organic waste, which are the inputs for the biogas plants. Organic waste include vegetable waste, fruit waste, meat waste, and cow dung. What are the microbes involved? The microbes involved are methanogens. And what is the catalyst used? We have used a catalyst here. The catalyst is dilute acid water. It was found that when we use dilute acid water with any of these organic waste, its efficiency for biogas production was improved. Each organic waste was used for a day, and its efficiency in biogas production in terms of the time period of availability of gases for the organic waste used was measured. So it took 3 days to use each of these wastes and another 3 days to involve them along with dilute acid water. We measured the time period of availability of biogas and made a tabular column giving the various values. Having obtained the time period of availability of biogas for the various organic substrates that we utilized, we now move on to the calculation sections. Here, I briefly explain how we can get the values of efficiency for each organic substrate that we utilized in terms of a parameter, time. The procedure is as follows. Let us consider a bucket that we utilized in our experiment with weight WB as measured by a weighing device. We add some organic waste to it of some weight WR. We now place the bucket with organic waste onto the weighing device and obtain a measurement of the weight. This observed weight is WO, observed weight. As we can see, the weight or the observed weight should be the sum of the weight of the bucket and the real waste weight. So the real waste weight can be obtained as WR equals observed weight minus WB, weight of the bucket. This WR is very useful in standardizing our value of time, which is used to measure the efficiency of the organic substrates. Let us consider that an organic substrate produced biogas for a time period small t, and let it be as organic substrate A. Now if you consider another organic substrate B with lesser efficiency in biogas production, this organic waste should be able to produce more, or it will have more time of availability of biogas than this organic waste, which will produce lesser biogas and hence will have lesser value for T. But if we use more of this organic waste B, then gradually the value of time T for this organic waste B can also increase, and ultimately the value for T for A and that for B can increase, and finally there may be a chance where the value for B will match or even override with that for A. Hence the weight of the waste is also taken into account while we consider the calculations for efficiency. Hence we define a variable capital T, which helps us in determining the efficiency of biogas production, where capital T equals small t divided by weight of the waste. This is the standard value of time which helps us in assessing the efficiency of each organic substrate. Hence, the efficiency eta is directly proportional to the standard value of time t.
The result was that cow dung along with dilute acid water gave the most yield in biogas. So this is the most economical substrate for biogas synthesis. So why did I select this topic, biogas plants? We know that the prices of LPG cylinders are increasing and this trend keeps on going unabated. If you do search for an alternative for fuel, do go for biogas plants. I hereby conclude. Thank you.